In this lesson, we're going to be looking at something called epigenetics. So the learning question associated with this is to think about how the environment can affect our genome. So as you can see from this image, we're going to be looking at how the genes that we inherit from our parents and the environment can affect the expression of those genes and hence our phenotype. These are the learning objectives associated with today's lesson. And you need to have read pages 238 to 241 of your textbook to have an understanding of the terminology. These are the key words associated with today's lesson. As we go through, we will unpack each of these, and you need to make sure that you understand them in association with epigenetics. So first of all, we're going to be looking at what gene expression actually is. Now we have talked about this before, and as you can see from this diagram, gene expression is simply protein synthesis. So how we go about protein synthesis is twofold. It's through transcription of the genetic code for a specific gene, and then we translate that mRNA copy of that genetic code, and we synthesize it into the correct arrangement of amino acids, which will then form a functional protein. Now, should point out that not all genes are expressed in living organisms all the time. If we think about it, every single one of our cells has a nucleus with the exception of erythrocytes, and that contains the full human genome. Now, not every single cell expresses the proteins that it can express. Some of these, we say, remain switched off because we don't need them to be expressed. So, for example, if a cell is destined to become a liver cell, we don't want to express any information for bones or muscle or hair. Now, I'm just going to take a moment to review the arrangement of the nuclear material in eukaryotes. Now we know that DNA exists as a double helix, which we can see at the bottom of this cartoon here, and that double helix is a large molecule. So in order to compact that up and to save space, that nuclear material is wrapped around a group of 8 histone proteins, and this forms a nucleosome. So the DNA, which is wrapped around those 8 molecules, is a nucleosome. Now these nucleosomes all coil up and condense into what we know as chromatin. Now chromatin is kind of like that unraveled ball of wool type stuff that we see inside cells that are not undergoing mitosis. However, when a cell is about to undergo mitosis, it will supercoil up into chromosomes. And here we're looking at a picture of a double chromosome, and that tells us that it's just about to undergo mitosis. So why are we learning about that? So we're going to be looking at epigenetics. And the word epigenetics actually means above genetics. So it's not the actual DNA that's changing with epigenetics. It's something above that. And what actually happens is modification of those 8 protein balls, those histones, that can inversely affect how we express genes and synthesize proteins. So we know that gene expression is linked to these histone proteins because we know that the DNA is wrapped around those proteins. And we can see here where these histones are unspooled, it kind of looks like thread on a spool. We can see that that gene is exposed. And that means that this genetic code can be easily read. So it's like opening a book. You can read the sentence, the page really easily if it's fully opened. Now, in this position here, we say that the histones have undergone acetylation. So I've given that a little positive symbol to show that yes, we can read that gene sequence and the DNA is accessible. On the other hand, we have something called methylation, and I've given that a little negative sign to show that no, we cannot read that gene sequence because these histone proteins are all spooled up and it's inaccessible. So we're going to talk about what this means for epigenetics and gene expression. First thing we're going to be looking at is acetylation. Now, if we look at this cartoon here, we can see that the gene is accessible and therefore we can fully transcribe and hence express it. So when we have acetyl groups that are added to histone proteins, which is what these little orange pentagons are, what that does is it relaxes the coiled up histone proteins and it unravels that spool of DNA and it allows the code to be read. So in acetylation, the attraction between the DNA and the histone proteins is reduced and it unspools that DNA so that we can see that gene sequence. So acetylation actually promotes the transcription of genes because they're available to be copied easily. How that happens is that our enzyme RNA polymerase, which is what mRNA nucleotides use, can actually bind to the promoter site at the start of that gene and transcribe that genetic code. Now if we look on the other side of that, when we see methylation, what methylation does is it inhibits gene expression. Now how it does that is it actually causes those histone proteins to coil up and therefore gene expression is inhibited. RNA polymerase cannot bind because you're unable to read this sequence. So where this actually happens mostly is at CPG sites or cytosine phosphorus guanine sites. Now they're called that because these little methyl groups, you can see the little M there, attaches to the phosphate that is on top of the cytosine nucleotide. So we can see a methyl group here and we can see a methyl group here on this ladder image of DNA. Now on the nucleosome we can see that a methyl group is here, here, here and here, and there's a few other places as well. So these methyl groups will inhibit gene expression and they are commonly found attached to the cytosine base it's called cytosine phosphorus guanine or CPG sites because of the base pairing and where on the nucleotide it's found. So I'm just going to summarize what we've learned so far. We know that chemical modification of DNA has been found to control expression in genes. So remember, the idea of epigenetics is above genetics. We're not talking about physical change to that genetic code. We're talking about a chemical modification that indirectly controls gene expression. Now we know that cytosine bases that have methyl groups attached to them will modify the DNA. And this DNA is said to be methylated. And what you can see in this cartoon diagram is that these little methyl groups stick up. So this line here is the DNA. It actually acts like a roadblock. And it prevents mRNA polymerase from binding to that DNA because these methyl roadblocks are in place. Therefore, we cannot express a gene. Now, methylation has never been found on active genes. So this is why we can say with confidence that methylation will inhibit gene expression. And here we can see where there's no methylation, RNA polymerase will easily attach to the DNA coding sequence and therefore a gene can be expressed because transcription is active. So when the gene is switched on, so let's say we know that a gene is destined to be active in a cell. So for example, if we think about making liver cells again, and we know that a cell is going to become a liver cell, but the DNA becomes methylated, which means it has these roadblocks, what that means is the gene would not turn on. Another way of saying that is that the gene has been silenced. Therefore, our liver cell may not function properly in this scenario. Now, to summarize, DNA methylation switches off gene expression. And this is because those methyl groups that project out from the DNA will actively block RNA polymerase from binding. So if we cannot transcribe that gene, the gene is said to be silenced. Now what this means is that the DNA isn't directly affected, but it just cannot be expressed. Okay, so that's the end of this short lesson on epigenetics. I would encourage you to reread those textbook pages. There's only 3 of them, pages 238 to 241. And to consolidate your learning for this lesson, I want you to complete all the questions in 9.3 found on page 241 of your textbook. Now, I have put up the answers to the textbook questions, and I would like you to mark your work using this. And if you have made any errors in your notes, I would like you to write out model answers and make sure that you highlight or underline key terminology. If you still feel that you need a bit of an extra explanation onto epigenetics, I've selected 3 really useful videos here that you can watch. The one in the middle is quite a short one. The science show video is a really good one. It explains the origin of epigenetics and the research behind it, and as does the Amoeba Sisters one. My order of preference would be the middle one, then the one on the left, and then the one on the right, but you can decide.